Okay. So today we will proceed from liver. Oh, I should share my screen. Okay. So today, structure of liver. We will see the structure of liver at first. The liver is the largest gland in the body, weighing between 1 and 2.3 kg. So it is one of the most important point in case of liver. In question point of view, that is, which is the largest gland of human body? So the largest gland of human body is liver, and weight of liver between 1 kg to 2.3 kg. In between this range, liver is weighted. Okay, then liver is located in the upper right portion of abdominal cavity beneath the diaphragm and on top of stomach, right kidney and intestine. So here location is stated. Now see the location here in this picture. The liver is located at the top of stomach, and just behind the stomach kidney is located. Both the kidneys are located left and right side of our spine, and this left kidney is located behind our stomach. So if liver is on the top of stomach, so liver should be also top of kidney. So liver is located at the right hand side of our body, right side of abdominal cavity, and on the top of stomach. Means its one portion is on the top of stomach. Like you can see here, the liver is separated by two lobes. You can see here the one lobe is just like on the cap, just like the cap of stomach. The right hand side of right hand side of liver is larger and left hand side of liver is a smaller, and this left hand side lobe of liver is just like cap of stomach. Now located in the upper right hand portion of abdominal cavity beneath the diaphragm. Yes, abdominal cavity means under diaphragm, and on the top of stomach, right kidney and intestine. Shaped like cone. Yes, it is like triangle in shape. The liver is dark reddish brown organ. Now the color is depicted here. Which color suits liver? That is reddish brown. Color of liver generally reddish brown. And the color if changes, if the color of liver changes, then abnormality is depicted by medical science. Now, there are two distinct sources that supply blood to liver, including the following. That are oxygenated blood flows in the form of hepatic artery, nutrition-rich blood flows in the form of hepatic portal vein. You can see here. Hepatic artery is depicted by red color. It is leveled here also, and this hepatic portal vein is depicted by purple color. So these two are the sources of nutrition and oxygen. Don't confuse by these two. These two are supporting, these two are the sources of liver. Like don't confuse, like artery is supplying oxygen and this vein is collecting deoxygenated blood from liver. Not like that. This hepatic portal vein is uh, giving nutrition to liver, right? Artery Hepatic artery is providing oxygenated blood, but this hepatic portal vein is providing nutrition to liver. So don't get confused. And now UGC NET is making their question level to a bit difficult. So keep in mind these things, these antic or these exceptional things in human body. Now, the next point is the liver consists of two main lobes. You can see here two main lobes. The right lobe is a bit larger and the left lobe a bit smaller. Both are made up of 100 lobules. Lobules means small lobes or smaller part of lobes. These lobules are connected to small ducts. Ducts means tubes that connect with larger ducts to form common hepatic duct. Means all these lobules are producing bile juice, and these bile juice are collected by this hepatic duct, and then the bile made by liver cells are stored in gallbladder, and some part when needed they are sent to the duodenum. Duodenum, please answer me, what is duodenum? We have already discussed.
It is a part of the small intestine.
Yes, very good. It's the part of small intestine, or we can say it's the smallest part of small intestine, or the first part of small intestine. Very good. So this bile juice is produced by liver and stored, collected by hepatic duct, and then stored in gallbladder, and when needed then it is released to duodenum, the first part of small intestine, via common bile duct. Now this is the anatomy of liver. You can see very well. If you want, I can zoom in. See very clearly where the color of liver, and it is divided into two lobes, and these two lobes are further divided into hundreds of lobules, thousands of lobules, and from these lobules bile juice is produced, and these juices are collected by common hepatic duct. You can see this hepatic duct is bifurcated into two. Means obviously bifurcated means two only, so bifurcated into two lobules, sorry, two lobes. So from these two lobes, this bile juice is collected, and they are collected to common hepatic duct. This green color duct is common hepatic duct, and this green color organ just below the liver is gallbladder. It is labeled here also, and it is stored here, and gallbladder concentrates the bile juice. Means the bile juice produced in liver is a bit watery or diluted, but when it's stored in gallbladder, then gallbladder makes it more concentrated, because for emulsification of fat, concentrated bile juice is needed. So that work is done in gallbladder only. So when needed, this concentrated bile juice is released to duodenum, right? Now, we will know how important liver is, or how important functions are done by liver. So functions of liver are production of bile. Most importantly, production of bile is done by liver, which helps carry away waste and break down fats in small intestine during digestion. Next is production of certain proteins for blood plasma like albumins, globulins, and blood clotting factors. Some coagulation factors are also produced by liver. Coagulation factors means blood clotting factors, means if any injury is taking place in our body, then blood losses by but blood loss also stops at certain point. Why this blood loss stops? Because our body is self healed, or our body has a very good mechanism made by divine god, that our body can heal itself. So what happens? Different coagulation factors are present in our blood. So after some time, these coagulating factors make clotting, and this clotting stops the flow of blood from artery or capillaries from inside to outside of body. So these coagulation factors are also, some of these coagulation factors are also produced by liver, and some blood plasma, proteins of blood plasma like albumin and globulin are also produced in liver. Next is conversion of excess glucose into glycogen for storage. If you people know that glucose is stored in the form of glycogen, and where it is stored in liver only. Glucose is the ultimate, means glucose is the simplest form of carbohydrate, or from where we can get a vast amount or a large amount of energy. So glucose, our body needs glucose every time. So this glucose is stored in the form of glycogen. When you will learn, or when you will study diet and nutrition, then you will study there that glucose is stored in the form of glycogen, and it is stored in liver only. And yeah, when we need glucose, then this stored glycogen is again transformed into glucose, and then we use glucose. And to balance and make glucose as needed, obviously. It is stored there, and when we need, we can use that glycogen by transferring into it glucose. Then, conversion of poisonous ammonia to urea. Urea is an end product of protein metabolism and is excreted in the urine. Conversion of poisonous ammonia to urea means our body produces so much of waste materials, and those waste materials should be excreted from body, and one of those is urea, that is made up of poisonous ammonia. And this conversion from ammonia to urea is done by liver. Then clearing of blood of drugs and other poisonous substances. Next is breakdown of erythrocytes and removing bacteria from bloodstream. Erythrocytes means red blood cells. Okay. Now this is also an important point in question point of view. Erythrocyte means RBC, or red blood cells. So remember it. Erythrocytes means RBC, or red blood cells. Now breakdown of erythrocytes and removing bacteria from bloodstream. Okay, this function is also done by liver. And next is clearance of bilirubin from red blood cell via bile. This is a long process, or a deep process, how bilirubin is secreted, how it is cleared. So all these process are also done by liver too. Bilirubin
makes our urine yellow, or bilirubin makes our feces or stool yellow. So this bilirubin is secreted from waste of red blood cells, and this bilirubin should be cleared from our body. If excess bilirubin occurs, then which disease takes place? Can you please tell me?
Jaundice, ma'am.
Very good, very good.
Excess amount of bilirubin causes jaundice. So clearance of bilirubin is done in liver.
When the liver has broken down harmful substances, its byproducts are excreted into bile or blood. Bile byproducts enter the intestine and leave the body in the form of feces. Do you guys understanding this? Means the waste material are divided into two from liver. One that is stored or mixed with bile, and another that is mixed with blood. And this bile is going to intestine, and it is excreted from intestine by stool. And another that is mixed with blood, and that is refined. Blood is filtered from where?
Kidney.
Answer me.
Kidney.
Yeah, very good. Very good. So our liver is clearing these bilirubin, ammonia, all these things are clearing, and this clearance, when done from liver, then it is mixed into two pathway. One that is bile, and this bile is going to intestine, and from intestine it is mixed with stool, and stool is excreted from anus. Another, when it is mixed to bloodstream, then bloodstream is filtered in kidney, and this kidney excrete through urine. Okay, this is the process. Now we have completed the glands, means accessory glands and alimentary canal of digestive system. Now we will study the carbohydrate, protein and fat digestion.
Now, first we will study about digestion of carbohydrates. At first, mouth means in mouth,
Mechanical digestion and then chemical digestion takes place. The enzyme salivary amylase or ptyalin breaks polysaccharides into disaccharides, that is starch into maltose plus dextrine. These are the forms of carbohydrate. Okay, we take carbohydrate at starch, then it is broken down into maltose and dextrine by salivary amylase.
then in stomach, the low pH in the stomach inactivates salivary amylase, so it no longer works once it arrives at stomach. Okay, when salivary amylase only works in the mouth and esophagus. And then when stomach, when the bolus is reaching to stomach, then the stomach juices, HCl, all these things are working. Then salivary amylase is putting down. And then in small intestine, chemical digestion takes place. Pancreatic amylase is secreted from pancreas into small intestine. The enzyme pancreatic amylase breaks starch into smaller polysaccharides in duodenum. Digestion is done by enzymes produced by enterocytes, the cells lining in small intestine. So this is the process of carbohydrate digestion. Okay, at first carbohydrate is starting digestion from our mouth by salivary amylase. That is, polysaccharides are breaking down into disaccharides, right? Like when you are eating a banana, Then this banana is having fructose in it. Then this fructose is broken down into disaccharides. Then when it is reaching to stomach, then the salivary amylase stops working due to low pH or acidic environment of stomach. Then when it reaches to in small intestine, then pancreatic amylase is coming here and it breaks down starch into smaller polysaccharides. In duodenum, duodenum digestion is done by enzymes produced by enterocytes, means the lining of small intestine. So, here, polysaccharides are further divided into monosaccharides. Like sucrose is divided or broken down into glucose plus fructose. Maltose is broken down into glucose plus glucose. Means if two glucose are combined, then maltose is produced. Then lactose. Lactose is broken down into glucose plus galactose. So these are the monosaccharides. These plus signs are this plus sign is combining two monosaccharides, and three sucrose, maltose, and lactose are disaccharides. You can remember this by one what we say that mnemonic. Sulema. Sucrose, lactose, maltose. Sulema. These are disaccharides. These are the important point in question point of view in from diet and nutrition also. You will also learn these things in diet and nutrition. So sulema is disaccharide, sucrose, lactose, maltose. And these are the monosaccharides, glucose, fructose, galactose. These three are monosaccharide. By the end of this process of enzymatic digestion, we are left with three monosaccharides that are glucose, fructose, and galactose. These can now be absorbed across the villi of small intestine and into bloodstream to be transported to the liver. Means what is the whole thing happening? We are eating a banana in our mouth and salivary amylase is breaking down the polysaccharide into disaccharide, and the salivary amylase is stopping its work in stomach due to low pH, and then when it is coming to small intestine, then it is further broken down into monosaccharide. And what are these monosaccharide? That is glucose, fructose, and galactose. So our banana is having fructose in it because it is a fruit. Okay, this is the complete process of carbohydrate digestion. Now, next is protein digestion. At first, protein digestion taking place at mechanical digestion at mouth. Then stomach, mechanical plus chemical. Let's understand. The stomach releases gastric juices containing hydrochloric acid that activates enzyme pepsin, which initiate the chemical digestion of protein by converting protein that break peptide bonds and convert large polypeptide into smaller polypeptides. Okay, this pepsin in stomach is breaking down protein into polypeptide into smaller polypeptides. Is larger polypeptides into smaller polypeptides. Now, in small intestine, the two major pancreatic enzymes that digest proteins in small intestine are chymotrypsin and trypsin. activates other protein digesting enzymes called proteases, and together these enzymes break protein to tripeptides, dipeptides, and individual amino acids. So, at first, polypeptide is broken down into dipeptide and tripeptide, and then by the help of chymotrypsin and trypsin, di and tripeptides are broken down into finally amino acids or by proteases. So, the end product or simplest form of protein is amino acid. So, these are the also important questions like what is the simplest form of protein? What is the simplest form of carbohydrate? Right? Means simplest form of carbohydrate is monosaccharide. Simplest form of protein is amino acid. Next, once the tripeptides and dipeptides are all broken down into single amino acids, which are absorbed into bloodstream by villi or of small intestine. Now, what are villi? Please answer me.
The small finger-like projections in the small intestine which help absorb the nutrients after they break down.
Very good, very good. Actually, it increases the surface area of the small intestine. Okay, that's why absorption is done very easily. Now, next is digestion of fat. Fat digestion takes place in small intestine only. Bile from liver contains bile salts that emulsify fat. Pancreatic lipase converts emulsified fat into fatty acid plus glycerol. So this is the simplest form of fat, fatty acid plus glycerol. So this fatty acid plus glycerol is absorbed into villi from small intestine. It is fat is going to small intestine, and this bile salt is coming to duodenum, and it is simplifying or emulsifying fat into fatty acid plus glycerol, and then small intestine is absorbing this. Okay, this is the end of our digestive system. Now we will have some question answer session, because I think you people forgot all the previous classes. A long gap was there. I am very sorry for that. I will be regular from today. Now please answer me. How long our esophagus is? Okay. Now answer me on chat box. Are you guys ready to type? Okay, you were taking time in opening your PDF of slide, right? No problem. You should remember all these after this question answer. That is why question answer is important. Now you can see the slide, but after this you should remember. So only 2-3 answers I have got till now. 2 answers. Where are other people? Okay. Sonia ma'am, Rajdeep sir. Okay. So, what is the largest gland in human body? What is the largest gland in human body? Very good.
I want this type of quick answers only.
Now tell me, what is the color of liver? What is the color of liver?
Very good. Very good. Now tell me, how much of gastric juice is secreted each day by stomach? How much of gastric juice is secreted daily by stomach? Very good. Now tell me, how many lobes are there in liver? Now, very good. I'm happy with your answers. Now, tell me, parts of colon. What are the names of parts of colon? Colon. What do you mean by colon?
It's a part of large intestine.
Yes, colon is large intestine. Now tell me, what are the parts of large intestine or colon? Very good, Rajdeep sir. Till now, only Usha ma'am has answered. Very good, Usha ma'am. You're doing very well. Okay. Answer of Usha ma'am is right. It is ascending, transverse, descending, and sigmoid colon. Right. Now tell me, how much saliva is secreted daily? Okay, good. Now tell me, fundus is which part of which organ? Fundus is part of which organ? Good. Very good, Usha ma'am, Kalishwari ma'am. Very good. Aarti ma'am. Okay. Now tell me, what is bolus? Or when we call food bolus? Very good.
Okay. So bolus is the mixture of food and saliva in our mouth. Now, please tell me the diameter of large intestine. Length and diameter both. Kali experiment, what is the length? Please type the length only, Kali experiment. Okay, very good. So the length is 1.5 meter and diameter is 6.5 semi. Right? You should always write the unit. Otherwise, it's not depicted well. Okay, 1.5, 6.5, it's not right. You should always write the unit. Okay. Now,
Okay.
yeah, no problem. So please tell me what is chyme? Hmm. Okay, a bit okay. All answers are not right. You people have just forgotten about anatomy class. I have shared all my slides, but you haven't gone through. Okay, no problem. Very good, Usha ma'am. Your answer is right. When food or bolus is mixed with acidic gastric juice, then it is chyme. The acidic soup in stomach is not chyme. It is gastric juice only. Chyme is not an enzyme and chyme is not bile. When the bolus of food that is mixed with, when we are eating food and it's going to into our mouth, then it is mixed with saliva, and then saliva mixed food is known as bowel, and this bowel is entering into stomach and then entering into acidic environment. When this acidic means acid is mixing with bolus, then it is chyme. Okay, now please tell me the gland which is endocrine and exocrine at the same time. Very good. Pancreas is that gland. Very good. So please tell me the no, yeah, the activities of digestive system. Tell me the activities of digestive system in systemic order or systematically. The activities of digestive system systematically. Yeah, you people are finding from slides. Where is it written? I can understand. Very good. Very good, Usama. No other answers is coming. Okay. So answer of Usha ma'am is right, and it's very good. I impressed from Usha ma'am. Okay, son, ingestion, propulsion, digestion, absorption, and defecation. Now, please tell me what is the color of pancreas? Fast color and weight. Very good, Kaliswari ma'am. At least you answered. Pale gray. Very good. Aarti ma'am, I have asked about weight, not its size. Yeah, very good. And Aarti ma'am has already answered about its length. Now, please tell me, um, absorption as food is taken place in which portion of digestive system? Yeah, very good. Very good, Sonia ma'am. A bit absorption is also done in large intestine. Yes, very good. So, yeah, all of you, very good. Now tell me, what is the term that uh, consists of sequential alternating waves of contraction and relaxation of alimentary canal? Means this wave is secreted by the contraction and relaxation of alimentary canal. You guys getting it? This wave is secreted by contraction and relaxation of alimentary canal. Now tell me, what is this movement known as? Very good.
Okay. So we should conclude the class now. Go through the slides well. We will start a new system or we will have question answers tomorrow. Okay.
Can you please share today's slide deck?
All the slides have been shared in the group.
Ones that you used today, ma'am.
Yeah, all the slides have been shared.
Okay, ma'am.
If you people don't is not having it, I will resend it. No problem. Okay, so I think we should conclude with prayer. Sit comfortably. Close your eyes. Upright your spine. Adapt Namaskar Mudra in front of your chest. Inhale deep and long. Exhale completely. Inhale the fresh air. Inhale the energy. Inhale the freshness. Exhale the toxins. Exhale the negativity. Exhale the tiredness. Inhale the fresh air. Be physically active. Exhale and be mentally relaxed. Inhale deep and long, and now start the prayer. Sarve bhavantu sukhinah. Sarve shantu niramaya. Sarve bhadrani pashyantu. Ma kaschit dukha bhag bhavet. Om Shanti Shanti Shanti. Now open your eyes, rub your palms, put the palms in your facial muscles, feel the warmth, feel the energy, and open your eyes with a smile. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am. Thank you, ma'am.